I am pleased to talk about my recent study on how modulatory neurotransmitter genotypes shape temporal organization of functional connectome dynamics and cognition. First of all, I would like to provide you some background of the current study by introducing our foundational work, which was published last year. In this work, we were interested in examining the heritability of several time-varying characteristics of functional connectome dynamics. To obtain these time-varying characteristics, we extracted 4 connectome states from over 1,000 subjects of the Human Connectome Project database using heritable Markov modeling approach. Using the subject-specific probabilistic state time course provided by HMM, we could infer which state is active for how long and how one state transitions to another. Therefore, we examined 2 temporal features. First is fractional occupancy, which describes the proportion of the time spent in each state. And second is transition probability, which is a probability matrix of transition between all possible pairs of states. In this paper, we also tested several spatial and topological features of the connectome states, but I will leave them out from today's presentation because none of them are found to be heritable nor behaviorally relevant. In our previous work, because the features were multivariate, we used novel ways to estimate heritability of these multidimensional features. For example, we created the multidimensional space for each feature and estimated the distance between the subject pairs. Here, you can see that this monozygotic pair have closer distance compared to the unrelated pair. As a result, ANCOVA analysis showed that monozygotic twin pairs displayed the shortest distance for each temporal feature, whereas unrelated pairs had the longest distance. In other words, the more genetically related the subject's pairs were, the more similar their temporal features became. The classic twin study model, or variance component model, showed that their heritability hovered above 40%, which means that about 40% of phenotypic variance was explained by genetic effects. Finally, using the canonical correlation analysis, we found that these temporal phenotypes had significant associations with cognitive abilities such as cognitive control, impulsivity, language, and memory. I hope this illustration can further provide an intuition of how genetic similarity affects the temporal phenotypes. The subject pair in the left panel, which is a monozygotic twin set, shows that genetically identical subject pairs display similar duration of time spent in each connectome state. They also display similar state transition patterns. On the other hand, the subject pair in the right panel are genetically unrelated to each other. As you can see from the pie graphs and the directed graphs on the right, they show this similar pattern of fractional occupancy and transition probability. So, beyond this established relationship between temporal phenotypes of connectome dynamics and cognitive abilities, recent studies have also shown that these temporal phenotypes are associated with psychiatric disorders such as major depression and schizophrenia. Further, a recent prominent theory has suggested that ascending neuromodulatory inputs play an important role in driving such connectome dynamics and cognition. Therefore, we focus on the strong possibility that neurotransmitter systems impact cognitive performance through modulating connectome dynamics. Specifically, we hypothesized that inter-individual variability in the genes or single nucleotide polymorphisms in the genes that are linked to neurotransmitter systems may contribute to such relationship. Therefore, we first aim to characterize the profiles of genetic polymorphisms associated with fractional occupancy or transition probability, respectively. Then we aim to examine whether the temporal phenotype-specific genetic profiles can be used to further explain the inter-individual variability in cognitive abilities. This figure is a simplified overview of our analysis pipeline. Everything in the panels A and B followed our pre-registered project pipeline. Panel A shows how we selected genes, imputed the genotype data from the Human Connectome project, and conducted the quality control processes. Specifically, we focus on the genes that include receptors, metabolic enzymes, ion channels, and transporters of all neurotransmitter systems combined. After all the quality control processes, we had a set of SNPs for each neurotransmitter system. Panel B describes how we built our phenotypes. These phenotypes include fractional occupancy, transition probability, and cognitive factors, which are the exact same ones used in Chapter 1 study. Finally, Panel C outlines the steps of the statistical analysis. I hope the next slide can help you better understand the analysis steps. The first step was finding out the specific combination of polymorphisms that best predict each temporal phenotype. For example, we had 43 dopaminergic SNPs after the quality control processes. We used a stepwise algorithm which adds or removes SNPs based on the model's fitness parameter to identify a subset of SNPs that best explains each temporal phenotype. For example, out of 43 dopaminergic SNPs, 5 SNPs were selected for fractional occupancy and 3 for transition probability. Then we added random genetic effects and covariates such as age, sex, and population structure to build a full model. This full model was tested against the base model, which contained only random genetic effects and covariates, to evaluate the validity and significance of the full model. I hope this illustration helps you see how genotypes of SNPs that are contained in the final model can affect temporal phenotypes. For example, we selected a single SNP here, which is contained in the dopaminergic model for both temporal phenotypes. Pie graphs, which represent fractional occupancy, show that subjects on the left with homozygous genotype of major alleles tend to spend more time in stage 3 and 4, the yellow and purple colors, and subjects on the right with homozygous genotype of minor alleles show even distribution of fractional occupancy. Directed graph, which represent transition probability, show that homozygous major alleles tend to make more transitions between stage 3 and 4, whereas homozygous minor alleles tend to make fewer tendency to transition between stage 3 and 4. Then we tested whether the same genetic models that were specifically built for each temporal phenotype can also be predictive of cognitive abilities. In other words, the final models defined at the first stage were refitted to each cognitive factor to examine whether temporal phenotypes and cognitive abilities share neuromodulatory genetic profiles. In terms of the full models for temporal phenotypes, we found that cholinergic, dopaminergic, and serotonergic models substantially outperform the base model at predicting the temporal phenotypes. The significance shown here are Bonferroni corrected for 8 models. Then the 6 significant models were refitted to each of the cognitive factors. As a result, we found that subset of the neurotransmitter models were strongly predictive of the cognitive abilities. The significance on the right are Bonferroni corrected for 24 tests. To summarize, our study provided the first genetic evidence on the link between SNPs and connectome dynamics features. There have been previous genetic work focusing on static or structural connectome features, but not on the dynamics features. Specifically, we found that different cholinergic, dopaminergic, and serotonergic SNPs predicted fractional occupancy and transition probability. Importantly, our findings suggest that the substantial heritability around 40% of the temporal phenotypes identified in our previous work may result from this collective impact of multiple SNPs. Further, our findings suggest the potential genetic link between the temporal phenotypes of connectome dynamics and cognitive abilities. Taken together, our work identified that specific genetic effects may shape connectome dynamics and cognitive performance through the regulatory impact of modulatory neurotransmitter systems.